Welcome to Wholistic Wisdom Live, Awakening Your Inner Healer. Today, my guest for the third time. I can't get enough of the interviews with Dr. Sabine Hazan, the one and only. She's my go-to person. For those of you that are not familiar with her work, she's a gastroenterologist, to me, the top gastroenterologist in the world, researcher, and clinical trial expert. Today's topic, oh, and by the way, she's also the founder of Microbiome, which for those of you that are not familiar, please go there, look at all of her work. It's incredible. Now, to give you a little bit of a background, first of all, welcome, Dr. Sabine Hazan. Thank you.
Thank you. Thank you for having me.
Yes. So today's topic is methylene blue. And here's why I want to talk about methylene blue. With the entire COVID pandemic that happened and long COVID, the number one search product on the market has been methylene blue. And of course, it caught me by storm as well about a year ago. And I was initially very hesitant because I tend to go for all the natural products. And for those people for people who are not familiar, methylene blue was developed as a first man-made synthetic drug or synthetic medicine in 1871. In 1891, it won Nobel Prize by, I think, Paul Ehrlich for use in, what was it used for? Malaria. It was used for malaria in 1891. And all of a sudden, lately, you hear a lot of information, data, supposedly this data coming out on that it builds your mitochondria, that it helps with brain fog, and data looking at Alzheimer's. So, of course, I became curious and I started taking methylene blue. A big part of me should have questioned it and said, well, let's test also my microbiome and see what happens. Well, I was taking it for 4 months. And then I reached out to Dr. Sabine Hazan, and I said, what do you think of this product? She said, well, let's test your stool. And there is some information that you have been seeing, and I'm so glad we looked at it. Of course, I stopped using it, but let's talk about what you are finding in your own clinic, testing people's stools. And then, of course, I'm happy to share my results.
Yes. Well, like you, I was asking myself, what is it doing? What is it doing to the microbiome? And then little by little, I started seeing a lot of patients that were on it. And at the same time, they had very low bifidobacteria. Now, of course, bifidobacteria is killed by multiple things, right? You're drinking 2 glasses of alcohol, 3 glasses of alcohol, you may wipe your bifidobacteria. You're taking some medications, antibiotics, you may wipe your bifidobacteria. So definitely there's a lot of things that, can kill your bifidobacteria. But when you start seeing, a population that is similar, for example, a population that is, vegan, that is, or carnivore, and you compare them and they're from the same region, right? So you're looking at like California, same region, and you're noticing, they're all taking one product that is similar. And you notice that they have very little bifidobacteria. You have to ask the question, is the product causing the loss of bifidobacteria, right? So I think that's probably what happened with methylene blue. So methylene blue is interesting because it's one of those products that we use as gastroenterologists to tattoo polyps. So like, let's say I have a big polyp that I want to refind, and I remove it, but I want to make sure it's completely gone. Then I will tattoo it with some methylene blue. And then a year, 2 years, 5 years later, I can go back to the colonoscopy and say, the area where I tattooed it is still clean, therefore the polyp is gone, right? So to me, a product that does that to the microbiome, to the colon, and stays permanently, I have to worry about what is it doing to my gut, to my microbiome. Here's the reason. When I do fecal transplant, the process of taking stools from a healthy donor and implanting it into an unhealthy, it's not just implanting, right? It's really, you have to kill the biofilm. You have to remove the whole biofilm. What accumulates on that biofilm, on that colonic mucosa, is all the stuff that we've eaten that is toxic, that is, full of dye, full of, pesticides, full of toxins, right? So if that all stays stuck on the surface of the colon, and I'm adding probiotics, then those probiotics are never going to get a chance to implant because of that biofilm. Now, the problem is killing the biofilm also kills your good microbiome. So it's not as simple as, hey, I'm going to go in there and take a bunch of pills to kill my biofilm to allow for the probiotics to grow. It really is, it's a process. And so when we do fecal transplant, one of the things I like to do, and Dr. Barodi I know does, is really getting rid of that biofilm and seeing the mucosa flourish again. I always say it's kind of like gardening. When you garden and you have a garden full of weeds, what's going to happen is you're going to need to remove the weeds, and then you're going to need to turn the earth and then plant your seeds. And then you allow for, sun and light and the proper water to come in, and your garden starts growing again with diverse plants. If you don't remove the weeds, you're never going to grow plants. If you don't turn the earth or remove that thick layer of, bad earth, you're never going to grow anything, because you've got that thick layer on top of the dirt that really is not conducive to growing or garden. So it's really like gardening is your gut. So I think when people think of, and I've certainly become very sensitive to this, right? Because now everything I put in my mouth, I ask myself, is this killing my gut? Is this killing my microbiome? Because what I've learned from the pandemic was when you do kill your microbiome, it takes a long time to regrow, to replenish, to reaccumulate those bifidobacteria. It's not very simple. I mean, it took me 1.5 years to grow my microbes. And now I can say, you know what, I don't need to take anything. I have my original, baseline of my microbiome. Yes, the microbiome changes, right? It changes every day. But the thing is, if you stay consistent, your microbiome should be consistent. I've been testing my microbiome for the last, ever since I started my lab. And, I had a baseline that was pretty consistent, the same amount of bifidobacteria, same amount of, Bacteroides, Firmicutes, etc. When the pandemic happened, when I started taking, a lot of vitamin C, I started noticing my bifidobacteria went up, because I said, well, what have I changed in my diet? I have the same diet every day. I'm like, I eat my cheese in the morning or my crackers, my protein meal at lunch. I'm pretty much like clockwork, the same diet and the same drinks. So to me, nothing that way changes. And my stress level is always at a heightened level, no matter what. So that doesn't change, right? But if something, if I do something like I take 10,000 milligrams of vitamin C, I'm going to notice something change in my microbiome. If I take a lot of vitamin D, I'm going to notice something changed. That was really the basis of the discovery for me, was really using me as the guinea pig and saying, okay, well, how do I protect myself from COVID? How do I do all that, right? So to come back to, analysis of the microbiome, it's research. We're looking at a moment in time of that microbiome, right? And we have an idea of what you're eating because you're telling us what you're eating. So now we have to play detective, and we have to say, why is your bifidobacteria so low? So then you have to do process of elimination. Well, it's not from the beef because, you're pretty organic. It's not from the alcohol because you're not drinking so heavily. So you kind of do process of elimination, and then you go, well, you are taking probiotics, and those are good probiotics. So technically you should be higher. Even though sometimes we see probiotics don't even do anything, but in your case, you're doing things right. So, methylene blue, we have to kind of pay attention. And here's the other thing that made me pay attention to methylene blue is I was treating patients, you know my protocol for gut health. So I was giving them my protocol. And me, I'm very strict with the diet, what you should be eating with foods, what you should not be eating. So I have my list of no-nos, and I'm very strict on that. And so I was putting all these patients on my protocol. And the majority that we would see, the bifidobacteria went from like 0 to increasing, right? Except for those people that were on methylene blue that did not increase. And that was the reason that I started asking the question, what is methylene blue doing to the microbiome? And it's, by the way, it's the same thing with hydroxychloroquine. Hydroxychloroquine is a great drug, was great during COVID at killing, at essentially killing the virus. But what is it doing to the microbiome? What is it doing to the bifidobacteria? Same thing with what we've seen with the vaccine. The vaccine, could be protective on some patients, but what is it doing to the bifidobacteria? And why the bifidobacteria? Because the bifidobacteria is that microbe we are born with as newborns. And as we die, we lose it. So, dead people, dying patients have 0 bifidobacteria. Cancer patients have 0 bifidobacteria. So the process of aging is really that loss of bifidobacteria. So, and also it's a $1 trillion industry of probiotics. So we really have to be very careful of what we're doing to the bifidobacteria and what affects it and what doesn't affect it.
Yeah, no, for me, as you said, I don't drink alcohol. I'm eating organic foods only, very conscious. And I was on a protocol of probiotics for quite some time. And then I've noticed, so initially when I started taking methylene blue, I've noticed, oh, I think it's doing something to my brain, like almost like a light bulb went off. But then I started having, after about a month, severe GI symptoms. And this is why it's like, what could it be? Because I started looking up studies online, but there's really no data online of what it does to the microbiome. And that's when I said, okay, I'm going to ask you a question because you are the one that's actually looking. No one else is looking. And I've noticed with several people that I've recommended methylene blue too. Of course, they've discontinued since then. Their GI has become a mess as a result of being on it as well. So absolutely. So now I'm tracking to see what happens to the people that were on it, that got off of it, including myself, to see how long it's going to take to rebuild.
Yes. And probably, it's probably going to take you at least a year, in my opinion, because, anytime you destroy something, it takes a long time to get back. It's like you break a leg and then you get in a cast. And then by the time, a year has gone by the time you can really go back to exercising the way you used to exercise. So I think everything takes time to heal, including the microbiome, in my opinion. And you're right, at the end of the day, we need to pay attention to that. We need to do the studies. We need to look at all these products. Maybe some people are doing better because it is killing some microbes, right? So here's the thing with microbiome manipulations. And, people always tell me, well, how should I improve my microbiome? Well, I don't know because I don't know what's living in your gut, right? If you've got a bunch of E. coli living in your gut that's overabundant, probably, we got to fix that problem, right? If you've got some lack of microbes, good microbes like bifidobacteria, we need to fix that problem as well. So I think, looking at it and saying, okay, well, this is what I'm seeing and this is what it is, helps in a way. Okay. But here's the thing. Once we discover that certain things are killing bifidobacteria, it's time to record them, right? It's time to say and I gave a lecture I did a podcast with Gut Girl, GI Girl, who's basically, she's the podcast for the gastroenterologist, and she brings up information on the microbiome. And we were talking about probiotics, and we were talking about, what's out there, what's to trust, and what are probiotics are doing, and which ones are good, etc. And one of the things that we started talking about was actually, this whole concept of telling patients that you're what's missing in your gut, and if that's valid, right? And that was one of, because it is research, right? And one of the things we started talking about was basically correlating it to reflux, right? And saying, well, remember when we discovered patients to have reflux, we discovered they had an inflammation in the esophagus. And then little by little, we started doing, testing where we did manometry of the esophagus, the G junction, and noticed that people with reflux had a weakened pressure around the G junction, which is the junction between the esophagus and the stomach. And then from there, we started realizing, well, what are the things that are affecting the G junction? Coffee, alcohol, fat, mint, but this took years to get to that level, to kind of get that list. So now as GI doctors, we can officially say, well, to our reflux patients, stop the peppermint, stop the caffeine, stop the chocolate, stop the alcohol. That took years to get to that. At some point, and you're seeing it a little bit with my protocol, which is a 3-page protocol, because I'm discovering things that are, changing, right? And that are killing the bifidobacteria. So of course that becomes on my list. Avoid this. Now, here's the thing. You have a urinary tract infection, you have strep pneumonia, you need that antibiotic, right? So that antibiotic is going to kill your bifidobacteria. But at the same time, you could say, okay, well, maybe I should do some vitamins and some bovine and some, probiotics to increase my bifidobacteria and start eating better and start all that. So, I think that's the most important thing when you look at, replenishing the bifidobacteria and not killing, etc. So we're the beginning.
You brought up something so relevant. There's a lot of people that claim to be experts at something. And I actually just thought about it because about 3 years ago, my husband was on this craze of like peppermint oil. And he literally created an ulcer. He was in agony every single night.
Yeah.
And you just brought up, because people say, oh, you can just take essential oils orally. Absolutely not. You can burn a hole in your esophagus.
Yes. And that's the problem, right? That's the problem. People that do not have the education, that have not gone to pharmacy school, that have not gone to medical school, that have never really done a biology class or to even know what a bacteria is or a virus, or to understand what DNA, what RNA is, right? And these people are influencing your health, right? Mm-hmm. So I live in Malibu, and people in Malibu all the time come up to me and say, oh, you should really try this it's great. You should give it to all your patients with constipation. And I look at that and I go, do you know what that does? I mean, this is not a good product, right? But you can't convince them because they're making money off the product. So when money is biasing your livelihood, when it's biasing your way of being, essentially, because you're just become a snake. This is what a snake oil salesman is all about. It's someone that's just a good salesman and can sell you crap. Now, yes. Crap doesn't heal anything. The good microbiome does, but crap doesn't heal it. So,
and that also goes for biohacking industry right now. Is such a huge industry, but most of them are not even looking holistically at a person. It's about what can you hack, which I have a problem with that. What can you hack in your system? You have to look at the entire system. It's not hacking one part, just hacking the brain or your mitochondria, but what does it do to the rest of the body?
And healing is the whole entire, I can't heal a patient if the patient does not have a connection with me, if the patient doesn't trust me. I have a woman who flies in once a month for me to help her with her husband who has Alzheimer's. Okay. And that woman trusts me, and I'm going to bend over backwards for this woman because she trusts me and I want to see the data if I'm improving her husband's condition. Right. So it becomes a patient doctor relationship. But here's the other thing. I bring more and she brings more to this relationship because it becomes a union, of two people wanting to help another person, right? Mm-hmm. So when you do that connection between humans, that love that gets, passed on to help another human being, is really important in the healing process. It's not just what I'm doing to the microbiome. it's really that confidence. It's really that whole, it's the spirituality, it's the belief, it's the, the wanting something so badly that, and that's what healing is. So the idea that we're going to decentralize doctors and remove them and replace them by a bunch of robots, well, yeah, the robots is going to tell you take your aspirin, or take your antibiotic, or you have cancer. But what is that going to do for healing? If I'm the patient, the last thing I want is a robot telling me that I have cancer, right? What am I going to do? How am I going to process that, right? I'm going to just, want to die, right? Because nobody's giving me that faith, that hope, that light. So a big part of being a physician or having healing is really that holistic approach, which you're all about, which is, yes, we'll give you the pills. Yes, we'll do it as safely as possible. But at the same time, I'm going to hold your hand and I'm going to have a relationship with you, and sometimes that relationship is not possible because people put barriers between you and them and don't let you in. And also, it's not possible because they're so reluctant. They're so, I don't want to say stubborn, but they're so, you have to let it in. You have to let the healing in. You have to let the belief. You have to have the faith and you have to trust, right? When you have that relationship between a physician and a patient. And I think because we live in such a society that's robotic in a way, we've lost that feeling. We've lost that people connection. And to me, that's the most beautiful thing, is really that connection. I have a mom with two kids that are autistic, right? And I'm helping her, and she owns a bunch of lambs and a bunch of goats and a bunch of cows and chickens, and she shows me pictures, 'cause she's trying to bring back her kids to health the natural way without having to do fecal transplant, right? So she's doing it very naturally, and she sends me pictures, and it's beautiful. And she shows me, like, look, he did this today, look, he's paying attention to his brother. So we're trying to change the path of a disease of two boys by changing the environment, by manipulating, by refloralizing their guts without necessarily having to do fecal transplant. And I really think that's the gold, right? Where you don't have to depend on a pharmaceutical product. You don't have to depend on a stool product, on a donor, where you can just depend on, hey, why don't I change my environment? Why don't I heal myself? Why don't I warrior up and figure out how to change it all, from my brain thinking positive to my, waking up every morning and doing my exercise to I'm going to eat the right foods. I think that's so important.
What I wanted to say, Sabine, is I think there's very few, unfortunately, there's very few medical practitioners that have massive influence around the world that are not just sitting around and shuffling papers and looking at what is being published, but are actually doing the work with integrity. And you are one of those people, because for me, you're my go-to person. And I just want you to know publicly that you have inspired me and changed the way I even view things in such an amazing way. That's why I think that any patient that comes to you, you're a gift to them. So of course the trust is going to be there. I can't imagine somebody not trusting you, because I know you come from a place of passion, love, integrity. You're going to show to the person what is, instead of some of the things that we see right now with this whole communities of hacking, which is marketing. It is not looking at the whole picture. And this is really, I think, the message from today, that my biggest message, and I think it's yours as well, is you cannot separate mitochondria from the microbiome. You cannot separate the brain from the gut. And that's really the key. If you're seeking for that one thing, it's never about one thing. It's
and mitochondria needs sugar, right? They need calcium. the ATP, the Krebs cycle is not going to work without sugar. You can't have sugar if you don't have the proper microbes to absorb the sugar to help in the absorption. So yes, it's all connected. That remember at the beginning of the pandemic when they said the vaccine just stays in your deltoid. Well, that was the biggest propaganda ever, and those were the biggest lies ever. Nothing stays in a deltoid. It's not like you have a little pocket in the deltoid and you sterilize it and it goes in there. The deltoid is supplied by nerves, is supplied by blood vessels. As soon as you put that injection, it travels the blood vessels. The blood vessels go to the brain, go to the heart, go to the lungs, go to the gut. In the gut, it starts settling there and it starts doing its thing. And then you pass it through your poop. I think the idea, and I give this example to people, the idea that something stays in one location is wrong. I learned that from the microbiome. You take a woman who gets Botox and she injects it in her eyes, you're going to find the microbe in the gut. Okay. So it is there. Where did she get it from? It came from here. That was injected because it goes in the blood vessels, travels, ends up in the gut. Eventually, part of her microbiome now. You know what I mean? So, everything we put, you have Staph aureus on your skin, you have it in your gut. You have influenza in your lungs, you have it in your gut. It's not going to just stay in your lungs. You have E. coli in your bladder, it's going to go in your gut. Everything passes, all these microbes, travel. And so that idea that, oh, well, we just got to focus on the brain. No, the brain is linked to nerves, is linked to blood vessels, to the gut, to the heart, to the lungs. It's all interconnected. I think when we separated the fields is when, and we stopped working together as physicians, is when we stopped, achieving improvement. Because, well, if it's not in GI, then it's no longer my problem. If it's not in the heart, it's no longer my problem. Well, whose problem is it at that point? You know what I mean? And then the primary care doctor can only handle one thing at a time. And his ability is basically, to refer to different specialists. But to connect all these it is very difficult. One of the beautiful things that I did with this pandemic was collaborate with a lot of specialists. So we discovered a signature microbiome for anxiety, and I called Dr. Sasha Bistritsky, and I said, why don't we start publishing on this? And now we're presenting it at the anxiety meeting. So it's amazing because here's a psychiatrist who sees anxiety, who treats anxiety, and here I have found a signature microbiome in the gut that maybe if we treat the anxiety with gut health, maybe we could decrease the anxiety, right? Same thing with Alzheimer's. I published a paper with fecal transplant where Alzheimer's patient started remembering his daughter's date of birth. Well, that opened up the world with Dr. Sheldon Jordan. And now we're working. He does what he likes to call hacking the brain. And so he fixes the brain and I fix the gut. So that's the protocol we're putting together. We have a Regenesis project that's going on. So I think the more, physicians, and it's not only physicians, it's scientists. I realized during COVID the importance, and having opened my microbiome lab. So I opened the lab really for me to understand the microbiome, to understand, how do we stay living longer? How do we avoid Parkinson's? How do we avoid Alzheimer's? Because what if I'm that patient with Parkinson's and Alzheimer's, right? So I don't want to be that doctor that says I coulda, shoulda, woulda done the research. I want to do it now. And this was kind of like my midlife crisis in a way, because it was interesting. The Woolsey fire for me in Malibu was a turnaround. It was the moment where my backyard, my house was caught. The fire was all over. And I was ready to lose my house. And I realized what was important was really bringing supplies to my neighbors and being there for my community. And that's when I really realized who I was as a human being. It was a test. It was a test that says, help your friend and your house is going to be fine, or help your friend and you're going to be fine, right? And so that was the big thing. But it was also an awakening for me at how little I can trust who I believe to trust, the government, that I can't trust the government to save my house, right? Can I trust the government to save my life? Who is the government? I mean, the government is only capable of doing up to a certain thing, right? That's why the private sector is so important, because we're able to do a lot more than what the government is capable of doing, right?
Yeah, it's innovation. This is where
Innovation.
innovation happens. Yes.
Exactly. And so it happens in the private sector, because I always say, yeah, I'm friends with people like the National Institute of Standards. And I always say, well, why aren't you writing the book of microbes and disease? They don't have the capability. They don't have the capacity. They don't have the funds. I mean, this is a huge enterprise to feed all these scientists. So it's not that they're not capable. It's just they don't have the ability to innovate because they're being told what to do. Like today, you're going to do this project because somebody's paying for it because they have the money to push a drug to market, right? So the government's going to basically support what is bringing things to market, and it's going to do due diligence in a way up to a certain point. Although, with the pandemic, we're all like, shaking our heads. But I think for me, what was so important was seeing with my own eyes, right? Why did I step into COVID? The same reason I stepped into the Woolsey fire, to help my neighbors. For COVID, it was different. It was basically like, I don't trust those people to do what needs to be done. I need to see it with my own eyes, right? When I wrote those protocols of hydroxychloroquine, Z-Pak, vitamin C, D, and zinc, and submitted them to the FDA, I did them. Yes, initially we thought, well, we could be a pharmaceutical company like everybody else. And then pretty soon, sometime in May, when I saw the censorship of my protocols, the censorship of advertising, I said, wait a minute, something is rogue here. Something has gone wrong. I've always been able to advertise for protocols for pharmaceutical products. I did psoriasis studies. I would put an ad on Facebook and bam, I would get 10 patients. In the middle of a pandemic, we could not recruit one patient on social media. We were censored. We were blocked. Sorry, cannot. And I have all my pages, LA Clinical Trials, Ventura Clinical Trials on Facebook. I have all these pages where I would always post an ad that was approved by the IRB, Regulatory Board. And every time it would basically get blocked.
Blocked, yeah.
And then my name started getting trashed on social media. And that's when I said, I'm a nobody. I'm trying to see the data. Why are you attacking me? I'm doing research, And that's when I kind of like started speeding up and started saying, okay, well, and really not trusting, right? I mean, by nature, I'm a non-trusting soul to begin with. They say the most paranoid people live the longest. So by nature, I don't trust. So, I come from Morocco. It's probably part of my culture, whatever, So to me, I don't trust. So if on top of that, you do things to me that is censoring me, I'm definitely not going to trust you. And I'm definitely going to do my research, and that's what the pandemic did to me. It really brought out the researcher that wanted to see the truth, not to publish, just to see the truth. And what I realized, because it's not about ego for me, I couldn't care less if I don't publish. Like people think like I'm upset if they criticize my article, like big deal, whatever. Okay, I know I'm right. Prove me wrong, right? So I don't care. And it doesn't affect me. I mean, it affects me a little bit because of course we're all humans, but it doesn't affect me to the point that I'm going to stop doing my research, right? If they stop me, then I'm going to, if I, for some reason, I can't do research tomorrow, I'm going to do everything in my power to hire 10 doctors to do research, right? So I'm not going to, the more they stop me, the more like I become like a hurricane, right? And so, and that's my personality. That's my personality. But what I did discover this pandemic that was beautiful is working with scientists as a physician. They see a side of the lab that they're convinced is the answer, right? But then I look at that side and I go, well, how is that clinically significant? And we start having these arguments between scientists and doctors. And that's where the beauty lies, right? Where you can talk to the scientist in the lab. For example, Kevin McKernan and Philip Buchholz, amazing scientists, they found contamination in the vaccines, right? And maybe that contamination could explain what I was observing on the front line, which was a loss of bifidobacteria from the vaccine. So one of the things that I basically asked them was, well, how is that clinically significant that you found this contamination? How do we go from what you saw as a contamination to what does it do? Is it actually killing the bifidobacteria? Is it actually presenting inside the human genome? Right? That's the next step. And nobody's going to be investing in that research, right? Because it takes good scientists that are righteous and good doctors that are righteous to do that research. But that's the step that needs to be happening, right? So the beauty for me, this pandemic was really connecting with the scientists and the specialists and coming together, which was what I envisioned anyways with Progenibiome. Progenibiome was really a company for doctors, for pharmacists, for healers to understand the microbiome together, to see, hey, something. So for example, back to Methylene Blue, Methylene Blue is working on some patients. Why is it working on some patients? What microbes is it killing? I had a meeting with Dr. McCullough. I don't think he ever posted it. And I told him, I said, let's look at it. Let's look at before and after. Take your patients that you're about to start Methylene Blue.
Yes.
Especially if you're so convinced that it's working, right? Take your patients, give them the Methylene Blue before, and then and then test their stools before, test their stools after, and let's see together what we're seeing, because that's how you accomplish a change, right? When we published Vitamin C Increases Bifidobacteria, it started with my case first, showing that, hey, I took 10,000 milligrams for 10 days and it increased my bifidobacteria. What is it doing on other patients, right? And then from there, what is it doing long term? What if I stop my vitamin C? Does it drop my bifidobacteria? Those are all the questions that we must ask as scientists and researchers. And
what did he ever say to you? Did he ever say he is going to look at the patients before and after?
No, he said, but his opinion is basically that we're giving too much. He thinks that it's a dose dependent Methylene Blue. He thinks that there's a certain level that you give. I
took a very low dose. I never told you how much I took.
How much?
So I actually took quite a low dose. I was doing drops. So I did about 3 to 4 drops. So that's about probably 2. I think that's very low dose. So I was on a low dose. And do you want to show? I'm going to pull up the slides. Yeah. So people can see. So again, I eat very healthy. I was on probiotics, really good probiotics, and following actually Dr. Sabine's protocol prior to getting on Methylene Blue, and while I was on it on quite low dose. So again, dose dependent. So here we go. Let me see.
So the first picture is really all your phylums. So you can show.
So, oops, let me. Is that this one, the first one?
Yeah, so that's your relative abundance. So what you see there is basically all the phylum that exist in your gut. And so you've got a bunch of phylums. The most important are the Actinomycetata, which used to be called Actinobacteria. Pseudomonodata is the proteobacteria, and then Bacteroidata is the Bacteroides. Bacillata used to be called Firmicutes. They changed the name. I don't know why. So those are really the most important phylums. But when we look at the bifidobacteria, it's in the Actinobacteria phylum, which is the Actinomycetata. So you can go to the next picture.
Is that it? This one?
Yeah.
Oh, hold on. Let me share. I don't know if you can see. Is it the gray one?
The second one. Let me see. It should say
this one.
Oh, no, that's okay. So that's looking at your Lactococcus. You're the person on the left that has 0 Lactococcus compared to one of my donors that has a lot of Lactococcus. So that's not present in you. So I wanted to showcase that. But
What
there
is that? What is Lactococcus responsible
well,
for?
it's you know, it's basically one of the microbes that you have when you're young. You know, mechanisms of calcium absorptions could be linked with that, you know, so Lactococcus is definitely should be present in a person that's perfectly healthy. So go on to the next one.
Okay, the next one, let me share. Let's see. New share. This one.
And remember, this is deep genetic sequencing. So this is your bifidobacteria. And you can see compared to a super healthy person, you're a little bit on the low side. The good news is you're not zero, but you're 0.51%. So that's a relative abundance of 0.51%. So remember, this is a DNA test, meaning that it could be alive or dead bifidobacteria. If we were to do what's called an RNA pipeline, that would tell us whether the bifidobacteria is alive and whether it's reproducing or whether it's actually killed. But you could see your level compared to somebody that's healthy, super healthy, is very, very low. You know, 0.51% versus 14% relative abundance is low, especially on someone that's taking probiotics and on the right vitamins, etc. And
Yes.
keep going. This is the phylum of Actinobacteria. So again, you can see my donor is like 14% and you're at 1.6. So that's really low.
And you would say that most of it is killed off by, it could be the methylene blue, correct?
It could be, it could be, it could also be, you know, I don't know the level of the probiotics, if it's suppressing your own ability to make bifidobacteria. So that's another question. But definitely stopping methylene blue, reassessing, seeing if that helps and improve, and keeping you on proper vitamins might be the answer. You can go to the next one. So the next one I actually showed was a very good microbe that you have a lot of. So
Which one? Is it this slide here, the orange?
no, keep going. That's your phylum. Go to the next one.
Okay, so the final one, let me find it. So perhaps 1 second. Let me find it. Let me see. New share. What do you see on my screen right now?
Still the phylum.
The file. Okay. So I'm going to 1 second to get to
It's
it.
by itself. It's like a box on its own.
Okay, I'm looking. A box. What color box is it?
Hold on, I will tell you. It's the one that is, oh, purple.
Purple. Okay.
The last slide. Yeah, there you go.
You go in my chat. Okay, hang
And it
on.
says Bacillobacterium.
Okay, the last slide. So perhaps this one. Let me share it. Is that, do you see it now? No.
Yeah. Yeah, that's the
Yes.
one. Yeah.
That's the one?
Yeah.
Okay.
You probably want to remove
Yes, everything else. Hold on, let me figure out how to do this. Okay, here we go.
Yes,
No.
so Bacillobacterium is an important microbe. We're going to be discussing a lot of it, but it was one of the microbes that we discovered that patients that survived COVID, not having COVID, had a lot of. So remember we published that paper, The Lost Microbes of COVID-19. So there was two of the microbes that were not found in severe COVID patients were bifidobacteria and Bacillobacterium. But in the high risk exposed to COVID but never got COVID, they had high Bacillobacterium and high bifidobacteria. So even though your bifidobacteria is slightly on the low side, your Bacillobacterium, you know, pops up to being at least healthy. So
And is this the bacteria that we are born with?
no, this bacteria you accumulate as you get older.
Okay. And is it
Oh,
based on
and
the diet
this is
or?
not, no, this is something that there's not much that's known on this bacteria, I'll be honest, except that we're noticing that it is very important in immunity. Also some mental health, lack that microbiome without saying which condition. But eventually we're going to have, we're going to think more and talk more about that microbes as we discover more about it. But what we discovered for COVID is that the severe patients lacked that microbe and bifidobacteria, and the exposed never got COVID had a lot of that Bacillobacterium or bifidobacteria. So, which was interesting because a lot of people will come to me and never had COVID, and their bifidobacteria is not exactly amazing, but they make up with the Bacillobacterium being amazing. So, and that's not something that you could easily culture, and it's not something you could put in a probiotic. So it's not a microbe that you could say, you know what, this would be a great microbe, but it's unfortunately very difficult to culture and to get. So, but you have a lot of it, so congratulations.
Good.
Yes. So
Okay, good.
as we
Thank you.
boost up your bifidobacteria, you'll be in
Yes.
great shape with those two microbes. The other thing that I didn't show in your sample is really the diversity. You had an amazing diversity. And that's probably from your travels, Egypt, and following where you go, and where, because you're eating foods from different places and you're bringing them in your gut. So regardless of, the methylene blue killed something. By the way, you stopped the methylene blue. You're feeling better with your GI symptoms?
Yes, definitely.
Oh, good, good. Well, and by the way, that's the GI symptoms that tell you, right? So, when your gut is, I was eating lima beans, and I don't tolerate lima beans. It's not part of my microbiome. Moroccans don't eat lima beans. So I was eating them and I was extremely gassy. That's my microbiome telling me, you probably should stay away from that. Probably because I'm lacking the microbes that are helping to degrade that.
You know what? It actually reminds me when we were in Egypt, I had zero GI issues. We came back to US and I'm feeling a little off, but I think I'm just having a hard time with food in the United States, period. And it's glyphosate, something they spray, even if I buy organic, I feel it.
I was just telling that to my sister today. I said we wouldn't need to take vitamins if the orange was pure and giving me vitamin C pure. But unfortunately, we are spraying too much, and the environment is not, conducive. You go to Italy, you go to France, you go to Greece, to the
Yeah, I don't feel horrible. Yeah.
and I'm the same way, by the way. I'll eat bread here and I'll get very gassy and bloated. And I think it's the flour is bleached. And when the flour is bleached, that probably kills my gut too. So, but then I'll go to France and I'll eat the baguette. I'll eat a whole baguette and I won't gain a pound. I eat a little morsel of baguette here and I gain a pound, So there is something to be said about the food in America, the mass consumption of food and the mass manipulation
Production. Mhm.
manipulation of food. I mean, pesticides. I never understood those because there's so many ways to garden without using pesticide, by just using the plants. You put marigold, you put, basil, you put different plants around, and then you could grow beautiful corn without having to spray to worry about other animals, right? Work the diversity of the plants. What's amazing is that you look at this pesticide that has a poison on the container, and you go, okay, poison, stay away. And then you
Mhm.
go through these farms and they spray. I mean, like, we're not talking like a little like spray here with like little glimpses of water. I mean, it's gallons just soaking the ground. You're basically eating, swallowing that poison. I don't even know why they're putting hazmat suits when they're spraying, because they're getting it all over themselves. I mean, that would be an amazing study to do, by the way, though, is testing the microbiome of those people that spray
Mm.
pesticides versus people that don't spray the pesticides, or the microbiome of a farmer that uses pesticide versus the microbiome of a
Yeah,
farmer that doesn't use pesticide.
that
I
would
have
be a
an
good one.
amazing farmer whose whole family is, they're like the Partridge Family. They're singers and musicians, and he's got the most amazing microbiome, and so do all his kids. So I had him build a little shack in his farm, and I said, "Can I just send patients for you to just refloralize their gut and drink, the milk from the cow, and go, shovel some cow poop, and just to regain your health again." But yeah, that's the problem. I think we're in a society where we've sterilized so much. Think about what we did with COVID, wearing masks and gloves and alcohol swabs, and all those medications. We've sterilized so much, and we're killing the environment, we're killing the oceans, we're killing everything. So I think, it's a problem.
Yes, so I would say, just to conclude our topic, well, we've discussed so many topics, but for everyone who wants to get tested, I know that they can reach out into your office, right? And it's progenobiome.com, is that correct?
Yes, they have to realize they're entering a clinical trial, so they have a big consent. Part of being in the clinical trials, the price is discounted because the foundation pays for half of it. And then those that can't afford it and want to be part of our studies, please put your name on the list. Every time we get funding from the foundation, we start calling those people to start testing them for free. So, those who cannot afford testing, put your name on the list with Progenobiome. So,
Yeah, so for everyone who has brain fog, who's on methylene blue,
yes,
or
yes, everyone.
anything out there, it's important to test the microbiome, and I absolutely encourage every person to have tested with your lab.
Yeah, we're basically conducting the research, so this is a good way to get research data. And also, listen, we give a huge consent to tell the patient, we don't know what we're doing right now. This is this is uncharted territory. This is a new space, and so we need to look at it. And this is, to me, that's the best way to look at it, right? Where the patient is informed that this is what all we're seeing right now is like a bunch of microbes that are kind of popping up. Eventually, it is. I have full, faith that eventually it's going to be, a test that's going to tell you you're at risk for having cancer, you're at risk for having Parkinson's. We're not there yet. I wish I could speed it up to be at that level so I could say, yes, I was right, to jump in on board of this. But we're not there yet. There's a lot of research that needs to be done. It needs the physicians to step in and basically start testing with us so they can understand what we're seeing. We're privileged that we're doing 57 clinical trials on the microbiome and diseases. So we really see the data, in vegan and carnivore, in, the person that lives in New York versus the person that lives in San Francisco, the person,
Amazing.
from different races. So we can always kind of compare with what we have in our database, and then compare. And the beauty of it is, once we do raise a lot of funds, for the research, is to take these microbes that we see with autism and look at the whole group of patients, right? So with autism right now, we've got hundreds of stool samples. Eventually, what I'm going to do is take all those stool samples, run a different bioinformatic pipeline, but also try to find new microbes that these people have in common, right? Because in the microbiome space, we've only discovered maybe 1,000, and out of that, we know maybe 200 are good or bad.
Mm.
There's still trillions of microbes that are out there. What if there's a common microbe that's just not named yet for patients with autism or Parkinson's? What if Parkinson's is a microbe that you could isolate ahead of time? I have an amazing case, this is why this is research, okay?
Mhm.
It's the grandmother has a certain microbe, which will remain nameless, and had kidney cancer. The daughter has the same microbe and has urinary tract infection.
Oh, wow.
And the newborn has the same microbe and is getting like a lot of colds, right? Now, imagine if this is the microbe that is passed on from grandma to daughter to child, and that you could take that microbe and stop it at the childhood level to prevent the kidney cancer in the future, right? That's the future that I want to see. I want to see that I don't believe it's just genetics. I believe the genetics, play a role with the microbiome, with the microbes. And I believe we pass on these microbes. I have microbes, that my great grandmother, had, I'm sure, because she survived certain diseases, and I have them in my gut, certain infections, and I have those bugs in my gut. So when you see that, when you look at mother and child, and you see the similarity between the mom and her child, my own family portrait, I know my oldest and I were identical, and we're identical in our microbiome. My little one and my husband are identical in their personality, identical in their microbiome, right? So there's something there. There's even my husband and I, because we've been married for 30 years, we're starting to look alike. Like we think alike. We're exactly the same person. Like I know what he's going to say before he even says it. Our microbiomes are so connected, right? And this is why I think with C. diff, using the microbiome of the wife or the spouse was always the best idea, because they're so what one lost, the other one has. And so I think that's the future. That's what fecal transplant opened. That's the future I want to see. So yeah, I mean, we'd love to, see what we see in everybody and understand its research and help us and support the Microbiome Research Foundation, because that's how we do it.
Exciting.
Yes, thank you.
Exciting. Thank you so much. I'll post all the links below.
Thank you so much. And thanks for having me.